But biotechnology solved that problem. You can take the genes from humans, right? And then clone the gene, put into the bacteria, and you can produce recombinant insulin in bacteria exactly the same as human insulin, right? So that's the example of the power of biotechnology in solving our problem in healthcare system. Another um, use of biotechnology, for example, for drug deliveries, even compounds like um, methamphetamine or ansamycin, very toxic, kill cell, not only cancer cell, but also normal cell. But using biotechnology, you can attach it to monoclonal antibody that only specifically looking for HER2 receptor. You know HER2 receptor, right? It's on the breast cancer, HER2 positive breast cancer. So you can deliver this toxic compound specifically to the cancer tissue. So it does not just go all, over your body and kill, all other cell and get a lot of side effect. So you can use biotechnology really to use this very toxic compound as a, warhead, and then bring it directly to the target of the cell. In this case, any cell that express HER2 molecules, that is the breast cancer cell, will be killed by the compound. And biotechnology in drug discovery, as I mentioned, we can modify the biosynthetic pathway of natural products, or you can use synthetic biology, or you can use genome mining. So that's another terminology. Maybe some of you are not familiar. I will introduce or explain more about those. So those are ways that we use, try to accelerate or to increase our success is finding new drugs or finding new bioactive compounds, right? So that's also give us an easy access to complex molecules. If you have learned chemistry in your school, of course, people sometimes synthesize molecules using chemical reaction. But if the molecule is so complex, it's very difficult to make. Require multiple steps, maybe 50 steps. The yield's very low. It's not commercially available, not commercially feasible, I would say. So, it's nice to be able to publish something like that, but cannot be used industrially. So, but biotechnology can help us get to complex molecules in an easier way. And environmentally friendly, because instead of using toxic chemicals, you use biological system, right? So environmentally more friendly, and then good scalability of the compounds. You can increase the fermentation tank, for example, and then can be patented as well, because in some other country like United States, if you isolate natural products, you cannot patent them. You cannot protect them because those are not your invention. That's a gut invention. It's already there. You just discovered them. So you cannot protect. But if you use biotechnology to make something new, then you can protect them because you can claim those are your invention. Those are the one that you produce. All right. So that's also commercially very important. So my group at Oregon State University have been using these technologies to try to find new drugs, right? To develop new compounds from natural products. So we use biosynthetic engineering, for example. So meaning we engineer biosynthetic pathway of natural products. So we study how natural compounds are made in nature. So it's not only isolating compounds, without knowing how they are made. We try to understand how they are made. And then once you know how they are made, then you can modify them. You know, you can, uh, yeah, you can change the system. Like if you do Lego, right? If you know you develop a Lego, you know how you make this car, and then suddenly you decide, oh, maybe I want to change the headlamp, maybe, you know, so you can do that. So, but you need to understand first how to make them, right? So that's what we do with natural products. We don't only isolate them, but we try to understand how they are made in nature. So we have published a lot of publication on that. I will discuss more later on. We use synthetic biology. So we make um, important compound or useful compound using biological system. Meaning we take genes from different sources, put them together in a platform, biological platform, to produce useful compounds. Chemoenzymatic approach. You can use enzyme, right? Some enzyme, they are promiscuous, meaning they are flexible. They can be used as tool to derivatize natural compounds, all right? So you can generate derivative of compound using enzyme. That's what we call chemoenzymatic. Right? And then genome mining. Basically, you try to mine uh, genome information. All right. So meaning you need to sequence the genome. You know genome sequencing, right? Human genome sequencing. You can sequence plants. You can sequence bacteria. You can sequence everything right now. And nowadays, I know in Indonesia, whole genome sequence cost is very cheap now. Well, at least affordable. Then if you discover some organism like fungus, yeah, jamur, fungus or bacteria, you want to know their genetic backbone or their blueprints, right? You can just sequence using bioinformatic analysis tool. Then you can really analyze, you know, the chromosome or the sequence, and then you know basically this organism can do what. So you can even predict what they can do, right? So this is an example how we do about this. So basically, we have a compound, say pactamycin. It's a very, I don't know if I can use this. Oh yeah, I can use. So it's a very complex compound is very active. It's a it's a very potent anti-tumor antibiotics, but has very low selectivity. It kills almost everything. Yeah, kill cancer cell, kill fungi, kill human cell. You can't use it as drug because it kills everything. So we want to modify this molecule, but to synthesize them is very difficult. So what we did, really, like I said just now, we try to understand how this molecule is made in nature by the bacteria. So we look at the genes that are involved in making this molecule. As you can see, each error bioactive compound. Like when you prescribe azithromycin or erythromycin, you should know actually many genes are involved in making that molecule. Tetracycline, the same thing. All right. So pactamycin also, many genes are involved. Each of those genes or enzyme with specific function. So if you have 30 different genes, that means you're talking about 30 different enzymes with specific catalytic function that catalyze step-by-step reaction to make the final products, right? From the small molecule to the very complex molecule. So that's what we did. We spent a lot of time understanding the biosynthetic pathway of pactamycin. So each round, meaning one enzyme, one protein, the function, they are different. to make converting very small compound like this to the very complex molecule. So that's the knowledge, that is required if you want to do this biosynthetic pathway engineering, all right? And then since we know the function already, then we started isolating or producing derivatives of pacamycin, the one that are not usually produced in nature. All right, so you can use this technique to make new compound with different structure, chemical structure. And some of them much better than the natural products. So as I mentioned to you just now, the natural products is very toxic, kill bacteria, fungi, malarial parasite, human cells. But the new compound that we produce, sometimes very small modification, suddenly no more antibacterial activity, but still maintain anti-malarial activity and very low cytotoxicity. So now you are able to change the pharmacological profiles of the molecule by changing the structure slightly. All right. So this is something that we are, as a health care provider, should understand. Even though some compounds, they are similar, doesn't mean their pharmacological profile exactly the same, right? Sometimes slight modification of the structure change the pharmacological profile. So for that reason, if one compound being approved by FDA or Balai Pom, and if you have another compound slightly different, you think, okay, must be the same. So that's not a good idea to have, to think about that. You need to go through the whole process, toxicity process, safety profiles, and so on and so forth. But what we learned very quickly, many of those bacteria that we believe very productive in making compounds, but when we grow them in the lab, they don't produce that many compounds that they're supposed to be able to produce. So if you look at their genome sequence, right, and then you analyze them, you realize actually each bacteria, they can produce so many compounds. Like this is Saccharopolyspora erythrae. This is the bacteria that produce erythromycin, has 27 biosynthetic gene cluster, meaning they can, by design or natural, they should be able to produce 27 classes of compounds. But when you grow them in the lab, they only produce 5, meaning we just don't know how to, to induce them or to grow them so that they can produce all the molecules that they are able to produce. So pretty much every bacteria that people have sequenced the genome and analyzed them realize, wow, if you look at their blueprints, they can make many different compounds. But when you grow them in the lab, they don't make that many because the environment maybe it's not really suitable for them to make those compounds. So that's what we call by genome mining. We mine the genomes to exploit that information to make new compound. So for example, this is Streptomyces spectrum, has 31 gene cluster, meaning they can produce 31 different groups. But when you grow them, this is a HPLC, show only 4 different group of compounds being produced. So that's require us to sequence them. So if you are working on, natural products, or you are trying to isolate bioactive compounds from organism, if you can find unique organism never been explored before, the first thing maybe you should just sequence the genome so that you know exactly what this organism can do, rather than just randomly collect organism and then you don't even know what they can produce, and then spend a lot of time trying to isolate the bioactive compound, characterize them, and so on. I think rather now, because like I said, it's cheaper now to sequence the genome of an organism, then you do that first and then try to understand what they can make, right? And then, yeah, using bioinformatic tools, now there's a lot of bioinformatic tools, right? You can go online, you can find them. And then the challenge is how you can induce them to produce drug, to produce bioactive compounds. Once you can do that, obviously you just have to isolate them and characterize their structures. So perhaps you have learned characterize chemical structure using NMR, LCMS, and so on and so forth, right? So what we were able to do in the lab also, we can change the, the yield, the concentration of compounds that are produced. So sometimes you have this organism produce something, the amount is so little, it's very difficult to isolate, very difficult to characterize. Then using genetic engineering, we can induce the production, sometimes 10 times more compounds being produced. Or you can just completely wipe them out so that they can produce something else. And another aspect is that give or open up opportunity for you to identify smaller products that usually you don't see in the wild type, but after you do genetic modification, you start seeing them. And then you can isolate them and then isolate new compounds like this, uh, inhibit heat shock protein 90 and inhibit the proliferation of HER2, um, uh, expressing cell. So and then some of the compounds is even stronger or better than the natural products that people have isolated before. All right. So this is another ways of getting a new compound. But apparently, natural product production is not as simple as that. It's regulated tightly by global regulation and pathway-specific regulation. If you study about cancer, perhaps you have studied about how this signaling pathway in cancer cell, right? MAP kinase and so on. So all are regulated very tight, you know. The same thing in bacteria that produce natural products, also they are regulated very tightly whether they are going to produce something or not. Like the environmental condition, for example, the concentration of oxygen, CO2, pH, temperature, and external stress, all those affect the production of bioactive compounds. So that's another aspect or another field that's, I think, very interesting to study, really to understand how those biosynthetic pathways are regulated, right? And then you can also try to induce them using very specific ways, or by knocking out genes or changing the biosynthetic pathway and so on. But also there are many non-specific ways to do it. For example, you just change the culture condition, or you add elicitor, or you do what they call by co-culturing, or you challenge the organism with by adding a different organism in there. If you add fungus into bacterial culture, maybe they tend to produce antifungal agent because they want to protect themselves, right? So they produce something to kill their opponents or to kill other organisms surrounding, right? So this just more recent work in my lab. I will share some of them. Um, uh, this related to how we are trying to induce the production of bioactive compounds. So this project is done by a graduate student in my lab, Lee Scala. So Lee is very talented student. She discovered a cluster of genes in bacteria that somehow involved in the induction of secondary metabolite or induction of natural products. When she knocked out these genes, genetically, she knocked out the gene, and suddenly the bacteria changed the production of bioactive compound. So she knows somehow this gene cluster produced something that regulate secondary metabolism, right? And then she decided to take the extract of that bacterium and put into a different culture called Streptomyces silicolor that produced this blue color antibiotics by adding this culture from a different from the first strain into a different strain, and she was able to induce the production of this blue antibiotics. But interestingly, when she take this extract, she called it magic potion, because she didn't know what is in there. But what she knows is that this magic potion can induce production of uh, antibiotics or anti-cancer drugs. Then she put that magic potion into a completely different bacteria. And as you can see here, compared to the wild type, this different bacteria that uh, receive this magic potion produce something completely different in a high yield. So this is a very interesting way. And so it is kind of unpublished data that we are still working on right now. We try to understand what's going on there. What is the compound that interesting to find out. And then using technology, like you can do comparative metabolomics studies. You can compare the metabolomics of those bacteria that are induced compared to the one that are not induced. Then you will know what compounds are produced, where are the difference between the one with induction or not induction. Another system regarding this is called the EBO-EDB cluster in bacteria and algae. So it's what's done by a postdoc called Dr. Wei Zhao. And so this cluster has been shown in bacteria to give the bacteria the ability to protect themselves. Apparently, bacteria is a food of nematodes. Do you know nematodes? Like, chatting, right? It's very small. So nematodes like to eat bacteria, apparently. Like eat Escherichia coli or Pseudomonas bacteria. But these Pseudomonas contain this cluster of genes in the genome, produce something that prevent them from being eaten by nematodes. So if the cluster still complete in the bacteria, so the nematode won't come to eat them. But if you knock out that genes, so this bacteria is no longer able to produce that protective molecules, and the nematodes C. elegans will come and just eat them. All right. So we found apparently this cluster somehow making kind of a effector molecule that induce the transformation of tryptophan to indole molecules. That this indole molecule really the one that repel nematodes, that make nematode don't come. Right. So it's very interesting compound. So another graduate student in the lab, Sam cluster. Now in gram about 10 years now ago, we discovered that um, fish, fish can produce sunscreen compound. Have you heard about that? But so apparently a lot of marine organisms, they produce sunscreen compound because in marine environment, a lot of exposure of sunlight, right? So of course they have to protect themselves. And we found genes, two genes in fish that apparently can produce sunscreen compound. And so, of course, if you want to isolate a lot of compounds, you don't want to kill a lot of fish, especially from the embryo of fish. Then we need to use biotechnology to produce the compound. So what we did actually, we cloned the genes. We took the genes out from fish. We cloned them, put into yeast, Saccharomyces cerevisiae, or put into bacteria, culture them, and produce the compound. So now you don't have to go out and collect fish anymore, but you just grow them in the tank, meaning grow yeast in the tank and produce the compound, sunscreen compound. So why it is important? Apparently, the sunscreen product that we use currently, yeah, all of them are synthetic compounds, like azobenzone, right? Or oxinoxide and so on. They are synthetic compounds. And the recent study found that those compounds, well, if you go to the beach, of course, you put sunscreen to protect your skin from skin cancer. But then you play in the water, and then it's being leached out into the ocean. And those compounds kill coral. They kill coral embryo. They cause coral bleaching. You know coral bleaching? Coral usually very beautiful, but then suddenly all of them just gray and white. That's called coral bleaching. So we contribute to the killing of coral, basically by releasing all these chemicals into the ocean. So, and another study found this sunscreen compound supposed to be on the surface of your skin, outside of your body, but they found this molecule in your bloodstream. Apparently, it's being absorbed into the body, and there is no real study really about the safety of those compounds in the body. All right. So obviously, many cosmetic companies and pharmaceutical companies trying to find alternatives. Their target really like natural sunscreen if possible. So, and we have been lucky because then we work on this area. So we have a lot of inquiries and interest in that area. So what we are able to do also, we can do synthetic biology. It's not no longer making the natural products, but also making completely new molecule by taking genes from different sources. So for example, we took one gene from this fish, then one gene from the soil bacteria, and two genes from this plant pathogens. Of course, we studied already. We know the function of this gene. And then we put them together into a system, platform, to be able to produce completely new sunscreen products. So that's the power of biotechnology, really. You can make useful chemicals using biological system. It's not using chemical reaction that are sometimes toxic. So this is another approach that we are able to do. And with this technology, then we set out to create a biotech, small biotech company, like a spin-off company from the university. And then after several years of doing that, the company is being obtained or acquired by another company that focusing more on sunscreen product or cosmetic products. So that's the way kind of a success story in academia. Because again, we are in academia, industrialization or commercialization, even though nowadays it's more and more emphasized, that's not our main goal, right? In academia, our main goal is to train new generation of student or physician or pharmacist so that they can be successful in the future. But nowadays, because of the um, interest of commercialization, of course, if you have some good products from your research, then you can try to promote them. And in this case, now being developed in a different company called Archaea in Boston. So just hoping in the next several years this can be commercialized. Then at least I will feel a little bit happy. Then some the work that we did in the lab is finally useful in the society. So, okay, in summary, so I told you already, natural products continue to be a main important source of drugs that we use in clinics. Um, Cutting edge technology has been very instrumental or helpful to be to make us more successful in developing natural products. And then biotechnology has made and continue to make significant role in drug discovery and development. And then by synthetic pathway, synthetic biology are those are powerful technique or method to discover new compound. And then finally, of course, understand how natural products are made in nature and how they are regulated is very important as well if you want to be successful in finding those compounds and create a system that can produce enough of the compound for you to work with. With that, I would like to thank the people who actually did the work. So these are my current group members. This is Sam from Indonesia. We have a visiting scholar from Universitas Muhammadiyah Semarang, and then a lot of students from Oregon State University, and also this formal postdoc and students, our collaborator, as well as funding agency that support our research. All right. Thank you very much for your kind attention. Hopefully it is useful, and I'm happy to answer any question that you may have. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Professor Taifo, for the very eye-opening talk about biotechnology. There are plenty of ideas, I believe, that you introduced to us about several sources of maybe previously synthetic substances, but now we are trying to find natural alternatives to it, or probably the research is already ongoing. For example, you mentioned about maybe finding the more nature-friendly alternative to sunscreen, or an alternative source of natural insulin, or whether it's going to be synthetic insulin, and other parts of biotechnology that I find very interesting, for example, about the properties and behavior of bacteria and algae and virus or fungi, and the defense properties of bacteria and algae. I would like to invite our audiences. Maybe we have, I haven't greeted them earlier. Today we have our dean, and we have our vice rector, and then we have our teaching staff from medicine, and also from public health, and also from pharmacy. We will probably open the question and answer session. Okay, for the first session, maybe for our dean, Dr. Humarianto, please.
Thank you, Dr. Mirna. Good morning, Prof. Taifo. Firstly, I want to thank you for your coming here. It's pleasant. I hope you enjoy coming to Indonesia, and will bring our lecturer to go with you in the postdoc program. As you know, my colleagues still have to improve their competency, especially in pharmacies. There's a two question, Prof. Firstly, as you know, a lot of my colleagues, especially lecturer from the pharmacists and their student, perform a lot of research in the herb. But as you know, there's difficulty about to clinical trial. A lot extensive laboratory and herb based drug is performed, but clinical trial often face significant challenge before this therapy can be widely accepted. So could you describe about this problem, particularly in terms of providing efficacy, ensuring safety, and managing standardization? It is the first question. The second is, you know, nowadays, the development of antibiotic maybe have a slow motion. Yeah, so there's uh, no antibiotic, new antibiotic discover. And besides that, antibiotic resistance have become global health crisis. Uh, could you describe us about how to creating urgent need to a new antibiotic to answer this problem? Thank you.
Right. Thank you very much, Adakan, for the very important questions. I think it's very valid question to us, and the answer can be very difficult. So regarding herbs, obviously, we know herbs has been used for a long time, for centuries, right? For centuries, jamu in Indonesia, Ayurvedic medicine in India, TCM in China, Kampoyako in Japan. So everybody used herbs for a long time. And then why then we still have problem now that special to get, positive um, approval to use herbs in treating patients in clinics. I think the very common problem with herb medicine so far, because the data is very scattered and mostly empirical data, has been used for many, several hundred years, but there's no real solid pharmacological data sometimes. The safety profile, toxicity, right? So those data sometimes lacking. I think the main problem really is standardization. How can we, because we are talking about a mixture of zillions or well, some hundreds at least of compounds, bioactive compounds in there. It's different from modern medicine, when you're talking about atorvastatin, or we are talking about erythromycin or chloramphenicol or whatever. They're single molecules and has been studied very clearly, all the mode of action, all the side effects, right, toxicity. As a physician, of course, then it's up to us to consider the risk and benefit, right? All of them has some kind of side effects, and then but the risk, if the benefit is better than the risk, then we say, okay, let's go, use it. But it's different from Earth because you are talking about hundreds of molecules, and you don't even know, their individual function, right? And then that's fine if you use a mixture, as long as the concentration of them exactly the same. But how, because these sources, as you know, if many of them, of course, from plants, and then plants produce secondary metabolites differently based on the season, based on the location, the soil, and so on. So no consistency. And, the data that you refer to use herbs maybe does not apply. They don't apply to the herb that you're using in clinics because it's coming from different sources. Like propolis, right? Propolis being used a lot. Apparently, different places, and de- depend on where the bees get their food, and then completely change, right? So yeah, stuff like that is a challenge, really difficult to deal with. Unless if you have a um, like a farm or a field that you grow this uh, variants of a plant, that variant meaning need to be precise variants, and then you do exactly the same way treatment, including harvest and everything. Perhaps you will be able to control a little bit about the secondary metabolites or bioactive compounds in there. And another problem also sometimes, this is because I also serve on uh, student risk committee, like co-promoter or something like that for doctoral students, because of the lack of analytical um, instruments. They just did the pharmacological study without checking the metabolites before that. So they just assume, oh yeah, I'm using, for example, sambiloto. Oh, this must contain this. But hey, the one that you're using may not producing or contain that. So you need to do analytical test first, right? Mass spec or whatever is needed to make sure that you have all those molecules. Then you do pharmacological activity test, at least that way. But without doing that, you just assuming. Assuming is not good in science. Right? And no doctor will use your product. You just assume. Um, so that's a problem for the country. The second question about antibiotics, that's true. So most antibiotics that we are using right now, they were discovered in 1940s, 50s, to up to 1970s. Um, More recently, almost no new antibiotics are discovered from nature because, again, the approach that I'm trying to emphasize here, if we keep doing the same way, isolating compound, so apparently those producing organism, they tend produce the same compound that has been isolated 50 years ago. And we are isolating the same one. We call it like low-hanging fruit, right? Low-hanging fruit, the one that easily isolated. So we don't get new product. And so that's the reason why people try hard to find new sources or do genome mining and try to induce them just to do many different things. And surprisingly, the newer antibiotics that are approved by the FDA, many of them are beta-lactam derivative, right? Including cephalosporin derivatives. Uh, but that's actually tell us, in addition to isolation, actually medicinal chemistry is very important because that's exactly the same backbone, same pharmacophore, but by the chemist is smart enough, of course, they are using computational design and so on to modify the molecule to make it a completely different antibiotics. For example, in penicillin group, we have this natural penicillin, right? Like penicillin G or penicillin VK, right? But of course, those are natural compounds. They are very sensitive to acid. You can't take penicillin G orally, right? But then people modified the molecule, adding amino group on the side chain. Suddenly you get amoxicillin or ampicillin. So amoxicillin you can take orally because it's stable in acid. Just doing some chemical modification change the admin or change the pharmacokinetics, right? Now you can take it orally. But then most penicillin and cephalosporin, they are more, the natural one is more active against gram-positive bacteria. But now we have penicillin that is, there are active against Pseudomonas, gram-negative bacteria, or cephalosporin, right? So even like cephalosporin fourth generation, active against MRSA. So that's MRSA. So that's another aspect, medicinal chemistry, changing molecules, also very powerful tool really to change. But what I was trying to say is, I thought, there's no chance for penicillin or beta-lactam antibiotic to be a new drug anymore. We have been using it almost 100 years now. But surprisingly, even today, more recently, new antibiotics that are approved, still beta-lactam derivatives. So that's, eye-opening. I think that's for the lack of the fact that we are not able to identify new, unique antibiotics. But another problem actually that we face, because antibiotics have been available for a long time, the regulation also very tight. It's not like anti-cancer. So if anti-cancer drugs, you have some kind of side effect, toxicity a little bit here and there, they still approve them. But antibiotics, you can't have anything really bad. A little bit side effect, that's enough for them to kill the, the products, because we have option, very, very serious or tight about regulation. The third problem is developing antibiotic is bad for a company. No company now, big pharma, put a lot of effort in developing antibiotic. Do you know why? Maybe because if you discover very good antibiotic, they said, oh, let's keep this as a final resort. Don't use it. From the company perspective, we can make money if nobody use our antibiotic. Just keep until you find a very bad resistant strain, then use it. Right? So from economical standpoint, there's no good reason to develop antibiotic if we are a pharmaceutical company. For that reason, like U.S. government, really, and Gates Foundation, they have this special fund to help people develop antibiotics. Do that. And then even maybe dactomycin, because dactomycin. The story of dactomycin also very similar. It was developed by Eli Lilly. Eli Lilly developed dactomycin and it's good antibiotic, but they stopped because, well, they said we are not going to spend more money and time here because it won't make that much money. But then a smaller biotech company, they bought this, they acquire, and then they develop it and then sell, get 500,000, $500 million revenue. That's not Eli Lilly. Eli Lilly, if you don't make million or $1 billion, they don't see it as a good, good product. That's why they develop, for example, statin, right, or anti-diabetic drug, those, or cardiovascular medicine. Those the blockbuster, the way how they make money, right? So if antibiotics they don't make money, then they don't want to do it. So many reasons, but the reason why we don't see, new antibiotics.
Thank you, Professor, for the answer. I believe now we have one of the representatives of our teaching staff from pharmacy. For any questions from our own teaching staff, please.
Okay. Thank you for the chance. Assalamualaikum warahmatullahi wabarakatuh. Good morning, Mr. Taipo. Thank you for Master Taipo for coming to the fifth JAMHESIC event in here. So the question is, I would like to introduce myself. My name is Ponda. I am a participant from teaching staff. So the question is, what are the major challenge of biotechnology in drug discovery? And the second question is, what do you think is the most significant development biotechnology in drug discovery in recent years? And the third question is, what do you believe are the critical skills or qualities that someone needs to succeed on biotechnology in drug discovery? And the fourth question is, we know that pseudomonas have an intrinsic mechanism for tigecycline. So is it possible to change pseudomonas that are intrinsically resistant to tigecycline? Cycline in Indonesia still has a high sensitivity rate. Thank you.
All right. Thank you very much. Could you please repeat the question number one?
Yes. What are the major challenges of biotechnology in drug discovery?
Thank you. Yes. So the major challenges in using biotechnology in drug discovery. So right now, because the science, the knowledge is still evolving, so there's still some challenges in terms of dealing with the system, because we are dealing with a biological system, right? Biological system meaning they are very different from chemical or physical system, usually very precise. Biological system always some differences, right? So even same group of bacteria, some bacteria can be genetically modified, the other bacteria cannot easily for us to modify, for example. Even as simple as maybe everybody know to do PCR, right? Even PCR sometimes works, sometimes doesn't work, right? Depending on the primer, sometimes the primer is not as good as the others, and so on and so forth. So just a technical system, technical problems like that still need to be improved. And then I can just randomly answer your question, right? So what are needed really for, like students or scientists to start using this biotechnology for drug discovery? Well, biotechnology really require knowledge in biochemistry, in my perspective, and biochemistry and molecular biology, right, genetics, and so on. So those are actually all of us have received that training or that education, right, by biochemistry. Sometimes it's really to know very precisely or to know about DNA, RNA, protein, right? And then the rest really is all about training in the lab. So it's nice, if you spend some time really working a project in a different lab that really good at doing this thing, because like that's going back to the same problem, that question, the number one question, the challenges. Yeah, because it's sometimes everything looks good on paper. On paper, oh yeah, you add this and that and that, but the result completely different. So that's a knowledge or experience sometime. And then another aspect that I realized very quickly when you do biotechnology or research in this field, you need to have very good problem solving skill, because every time you do experiment, is all the results sometimes not the same, or you get failed results, and how you are able to solve that problem. Because on paper, everything looks good sometimes, all right? And then there are many example of success story about biotechnology, right? Even when people making I think one of the statin drugs, right? The statin drugs, and so that you have this pharmacophore and then side chain, and that used to be attached using chemical reaction. But the problem is the side chains alone already require several steps of chemical reaction, and then to attach it and then deprotect and so on require more step chemical reaction. And this is a story of my colleagues at UCLA. So they discovered an enzyme, that can specifically put the side chains to that position. So that's really shorten the the step of making the compound. This is commercially available. If you can shorten even just 2 steps, say 2 steps, that's a lot of different already, right? I mean, originally maybe you spend $1 to make 1 milligram. Now you only spend 50 cents to make 1 milligram. That's 100% gain. So that's very simple one. But there are many, RNA vaccine, that's biotechnology, right? And then there are many now, like monoclonal antibody. Nobody using polyclonal antibodies anymore, almost, right? So everything is monoclonal antibodies because it's very specific, and then this require cloning of genes, and then recombinant production of protein and so on. Those all biotechnology. So yeah, especially now, I mentioned at the beginning, we are entering the era of biologics, right? All these growth hormones, all the protein drugs. Yeah, those are products of biotechnologies. And yeah, and in the case of drug discovery, right? Drug discovery, nowadays, a lot of people, some people using different biosynthetic pathway and then transfer them. For example, the original bacteria does not produce enough. Then you can just clone the whole pathway, put into a different bacteria, and then you produce it different. Especially now, people pushing of making plant products, plant products like artemisinin, right? Anti-malarial drugs. So artemisinin story is very interesting. Perhaps you heard about Professor Yu Tu that got a Nobel Prize in 2015 because she discovered artemisinin as a new anti-malarial drug. We have been using quinine or quina, right? But nobody uses quina anymore. And then we have chloroquine, and then a lot of bacteria, a lot of plasmodium become resistant to chloroquine, right? Atovaquone, and so on and so forth. But artemisinin has a very unique mode of action. So that was introduced, but of course we don't use it as a single drug anymore because we're afraid that Plasmodium falciparum can become resistant to the drug very quickly. So you only use them as a cocktail or together with other antimalarial drugs. Just like when you treat TB, right? You have a cocktail of rifampin, INH, isoniazide, ethambutol, and so on. So, um, so, but the problem with artemisinin is because it's derived from plants. So a lot of farmers grow the plants and then they produce artemisinin. Sometimes overproduction and the price drop. Right? And the next year, farmers said, "No, we don't make money." Right? They stop. And then suddenly the availability of the compounds low and the price going up. So the price is up and down, it's like crazy. So what people try to do really to take the genes from the plants and put them into yeast and try to make artemisinin in yeast. Yeah, so that's one way. Another effort that has been, currently has been doing is to produce morphine or, you know, like uh, codeine and morphine in yeast. Because nowadays, so far we collect all those, get the compound from erythrocyan, from Papaver somniferum, right? So this cocaine, not cocaine, but uh, morphine field, right? So a lot of plants and very difficult to control. Now what they are trying to do really to get the genes put into the yeast so you can control the production in the uh, in the factory rather than having this huge field of Papaver somniferum field, so yeah, that's another direction. And people also trying to, like I said, use the system to make biofuel, right? To make short chain fatty acid or the one that can be used for energy, for example. So yeah, I think the emphasis, even in the US, is very strong on biotechnology moving forward. And then about Pseudomonas, how I mean, in the lab, we can change the resistant Pseudomonas against is that what was that the tigacycline, right? Tigacycline. In the lab, we can change that because Pseudomonas perhaps become resistant to tigacycline because they change their the ribosome, for example. Because tigacycline or tetracycline group, the mode of action, the target is a ribosome, ribosomal subunit, 30S ribosomal subunit, right? So that's where they work. They stop protein synthesis. So bacteria become resistant in a different way. One way is a target modification. So they modify the target of antibiotic so that the antibiotic cannot kill them anymore. Or they have efflux pump. So they create a pumping system so the drug cannot accumulate in the cell. They get in and then kick out again, So in the lab, you can study that, right? You can compare the ribosome between the sensitive and the resistant one and see if there are any different that you can identify that cause the resistance. If that's the case, then you can flip it back, right? They become resistant again. Or if they produce efflux pump, maybe you can find something to reduce the expression of their pumps. If the bacteria also can produce enzyme to modify antibiotics, right? If you use aminoglycoside like kanamycin, neomycin, bacteria can produce enzymes like acyl transferase or nucleotidyl transferase that can modify our antibiotics, so it is no longer active. Now, if you know that is the mechanism of resistance, then you can also change. But of course, you cannot control the one in the clinics. The one being passed through by, from one person to another or from nature. You cannot do anything about it. If they are resistant, they are resistant. you only find them after they infect your patient, right? So your patient gets sick, then you say, oh yeah, this is this. So does that answer your question? So yeah, technically you can change that, become resistant again, but I don't know if that is relevant at all in the clinical setting. Maybe. All right. Thank you very much.
Thank you, Professor. I'm sorry, I need to be a little mobile to the tables because I think plenty of our participants here, teaching staff and students alike, are all very interested. But probably they are still gathering, they're still arranging their questions for you. So I will try my best to organize them. Now, I believe we can give the chance to our pharmacy students because I believe we have a very important to leave this opportunity unattended. I'm going to give a chance for, as this is Friday, maybe one from this table and then one from this table. Untuk menggunakan bahasa Inggris atau bahasa Indonesia dipersilakan. Yang penting untuk kali ini ada bicara dengan latar belakang dan pengetahuan. Baik, terima kasih. Silakan pharmaceutical students.
Student in Faculty of Medicine and Health, Jambi University. It's my pleasure to meet you today, and I want to ask you something about interesting the development of the biotechnological, and I was curious about how the challenge and potential risks we need to consider in especially in pharmaceutical in Indonesia for innovation development, especially genetically engineered in synthesis and untyped natural material. Thank you, Professor.
Right. Let me answer that question first so that I don't forget. All right. The challenge in farmers, I guess the question is about the challenges in pharmaceutical student training in Indonesia, perhaps, and to, or in the case of research in drug discovery and so on in Indonesia. So, well, since this is related to education, I want to make a comments a little bit about our education here. So, in the U.S., so slightly different, right? Slightly different. So our pharmacists meaning almost the same as apotheker here, right? So the trainings really 4-year undergraduate plus 4-year for as a PharmD. So the degree is Doctor of Pharmacy. So the undergraduate level can be anything. You can be, from the Department of Chemistry or Biology, because we don't even have Department of Pharmaceutical Science or Department of Pharmacy. So as long as you get all the basic science courses, and we have this list of courses we call pre-pharmacy courses. Pre meaning before pharmacy courses that you need to take. If you pass them, then you can go to the pharmacy school. Pharmacy school require 4-year undergraduate education, and then you will have to spend another 4 years to get your degree before you can practice in the pharmacy setting, like community pharmacy, apotek, rumah sakit, and so on. All right. So 8 years. But we also have a program that toward research. So that's toward clinical work. So you cannot, well, in here it seems to me like, both research and clinical pharmacy come from the same place, right? Then you get a apothecary degree, but you still have to do research. Sometimes you do research in basic science. So over there is a step. So you have to decide whether you are going to work as a clinician, meaning pharmacy in the clinics, or you're going to work as a scientist. So that's a master degree, PhD degree. So you don't even do anything in the hospital. You just do research, basic science research. So it's more focused. So I think if we really want to, as a country and as a community, pharmacy community want to produce manpower, right, SDN, to do research, meaning good research, then the education needs to be very specific. Because I got my, degree, pharmacy degree and apothecary here in Indonesia. We know a lot of things. We know many things, but we only know the surface. We don't really go deep, right? So we thought deep knowledge, you can't really do much or good research, right? So yeah, that's part of the challenge, I think, the time that you have to get training in the field that you want to do, especially in research. All right. Does that answer your question?
Okay, allow me to allocate maybe about 2 more questions from students. I hope we still have time. Mungkin satu pertanyaan lagi dari farmasi.
Introduce myself. My name is Natania Panjaitan. I'm from Pharmacy Sudan. And my question is, the first, some of us did their final project by selecting plant extract and then do active bacterial activity test. And we find out the compounds that have this activity. And I want to know what needs to do next to so that it can be consumed by humans. Like what we found is can be consumed by humans. And my second question is, what do you thought for now is better to develop synthesis drugs, or it's better to make drugs from natural, like plants or animals?
All right, very good question. Thank you. So perhaps you learn maybe in class, the process of, from the discovery of the molecule until it's being used in the clinics. That with very intensive research require 15, 20 years, right? And with many different steps from the preliminary discovery, right? And then the next step will be clinical. Preliminary discovery, including, of course, pharmacology, isolation, chemical structurization, pharmacological tests, and so on. But you have this preclinical study as well, to learn the mode of action, to learn the toxicity, and so on. And after that, then you have to push it through the clinical study, right? There's phase 1 clinical study, phase 2, phase 3, and so on, before finally you get approved. So just to give you perspective, if you have 100,000, all right, 100,000 compounds you discover, all right? And then after this 20 years and costing million of US dollar, only 5 compounds being used from 1,100 compounds. All right, so meanings the chance is very low. Right? And then the problem also, and then obviously we in academia, we cannot do this clinical study, just too expensive. You need to have pharmaceutical company to get interested in your products, right? Before then they decide, okay, this is really, really promising. Okay, let's invest money and do the studies. If not, whatever you discover will, stay there. So nobody, if they are not interesting, then nobody excited about it, and then you can do anything about it. So that's what they call by the valley of death in drug discovery. From the beginning, there is a gap, a place where nobody wants to invest money. But if you manage to push your project until you pass this valley of death, then you get good candidates. Then maybe some pharmaceutical company would be interested and pay for all the study that are needed to push the product to the market. And of course, after it's being released, there are another study as well, post-marketing, you know, um, studies basically. Uh, so yeah, that's part of the reason why I always say, in academia, um, we the main goal really to train student. So what you're doing right now is very important. Meaning you get training, right? Training how to isolate compound, how to do this, how to do that. So hopefully you use that skill, and then you go out there, and then you can work in drug discovery, pharmaceutical company, discovery division, and so on. So you have this skill, right? But if you think like you want to bring whatever compound that you isolate now to be a clinically used drugs, that big challenge. It's a long way to go, right?
Thank you very much, Professor. I believe we have two more questions from the medical student table, maybe one from this table and one from this table. Who wants to go first? Maybe one or two questions from this table and maybe one or two questions from this table. Hopefully we still have enough time. Yes, maybe for the last session questions.
Thank you. Assalamualaikum warahmatullahi wabarakatuh. My name is Faisal. I'm a medical student from the Faculty of Medicine and Health Science, Universitas Jambi. Before that, thank you, Professor Taifo, for the presentation on the natural compound for the antibiotic drugs and sunscreen. My first question is there any clinical trials on human subject for this antibiotic drugs and sunscreen with natural compound for the resources? And if there is there any side effect on the drugs or sunscreen that we as the doctors need to be aware about? That's for my first question. For my second question is regarding the environmental issue. Using the natural resources means that we are dealing with environmental issue. Is there any effort on this resource that may be used for this environmental issue? Maybe that's for the questions. Thank you, Professor.
Thank you. Very good questions. So the first question about clinical trials of the compounds that we generated. So the only one that has been done with humans is the sunscreen compound, obviously, because it's done now by a company that we created, and then we got some funding to do it. And sunscreen, that's another thing, perhaps people don't pay attention too much. You depend on your product. Sometimes it's getting very difficult, sometimes it's getting easier. Like I mentioned to Pak Dekan just now, if you want to develop antibiotics, so the regulation is very tight, need to be very safe, right? But if you develop anti-cancer drugs, Well, a little bit side effect. Maybe they still consider it's okay because the need, because we need anti-cancer drugs much more than antibiotics. Although that may change after a lot of bacteria become resistant, right? Um, so, for sunscreen compound, is because it's external use. So it's the regulation a little bit less. Then you can test them again to humans and then get the SPF number, right? You know SPF, sunscreen always come with SPF. So that has been done. So now well, I say we, but actually the company has been acquired by another company. So they were able to scale up the production. That's another aspect in industry. Even though you have a very good compounds, right? But if you cannot produce enough to be used, that if they have to sell one bottle of sunscreen for say 5 million rupiah, nobody will buy it. So you need to be able to produce enough so that you can sell them cheaply. So that's about the products that we we tested for for well in human or on humans. And about the environmental issue, um, so depend on countries, right? So the regulation they're all different, and the U.S. has been very tight for the past maybe 15, 20 years. There's no new sunscreen compound has been approved. And then the one that is being used, or the one that are being used right now, they were approved many, many years ago. And then the knowledge about their effect in the environment, and even now they are, people know they are being absorbed into the body. Those knowledge just very recent in the past several years. But nevertheless, because of that knowledge now, many places they change the regulation. For example, like the state of Hawaii, Hawaii, right? So they ban some of the sunscreen products that they believe cause problem to their, coral reef, right? The environment in Hawaii, Key Peninsula in Florida, Australia also looking at, that possibility of banning protect some products to protect their coral reef. But so far, no response or no regulatory change related to the absorption of the compound in the body. Maybe the amount is little enough, it won't hurt anybody, but we don't know moving forward. But there's some aspect of that. But nevertheless, I think environmental problem a little bit difficult because it's kind of depend on how country perceive about that problem. Some even don't care. Some even they have a bigger problem in terms of environmental problems, right? So they have like plastic problems, in the ocean and so on. So yeah, it's difficult to help. But um, some places have take action really to try to protect them, from the negative effect of synthetic sunscreen compounds, right?
Thank you, Professor. We have maybe one last question from the student table. Or two. One or two.
Oke, Assalamualaikum warahmatullahi wabarakatuh. Excuse me, I'm Mutiara Dwiza from the medical student from Universitas Jambi. I want to ask, based on the presentation, for medical student like co-assistant, in the particularly in the clinical phase, who are closely related to pharmacology, what the steps, where should we start if we are interested to research on the pharmacology? but conducting research in pharmacology, and especially if our background is not from the pharmacology. What the action can be taken is the question. Thank you.
All right. Thank you for the questions. So of course you have options, right? If you are really interested in doing pharmacology, you can always, learn something, learn new thing. You can go to the pharmacology lab and then study or, get training there. And sometimes I have to choose, even in our case, right? In our case, when we discover something new, for example, we have to test based on the literature. Maybe this type of molecule has some kind of specific pharmacological aspect or activities. Then in my case, I have to choose whether I want to develop the assay system myself in my lab or just collaborate with people, collaborate with somebody else who are expert in the field of pharmacology. Then that way, first, you don't have to invest a lot of your time and resources to develop a new system. At the same time, then you get somebody helps with really, a lot of knowledge and experience already. But if you're young, you still, you still don't know much about where your future, where you want to go. Learning different thing is a good thing. I experienced it myself because I was trained as a pharmacist, but then, working in natural product isolation, and I did cytotoxicity assay myself. I did organic synthesis. So like total synthesis of natural products and do genetics, enzymology, molecular biology, and so on and so forth. So of course, it's not like overnight. Took me like maybe 10, 20 years to gain all those experience. But fortunately, just to be honest, I'm more comfortable now talking about all those things because I've done myself. It's not like I'm reading, just based on reading. So having your experience working in the lab, in the field, for example, you need to test with mice, you do it yourself. I think, yeah, depending on your age and career stage. For me, maybe, I'm not interested in learning something new anymore dramatically. Of course, I still read, I still do
Jawa, Jawa. Jawa, Jawa.
Backgrounds that involve pharmaceutical therapy or treatments in our field. Thank you very much. We apologize for all of the technical difficulties. We hope that didn't hinder the talk. And then now I would like to return the floor to the MCs and then to the organizing committee. To all the participants, maybe do not leave the meeting yet because we still have other agenda for today. Untuk panitia dan MC, saya persilakan.
Thank you for our keynote speaker, Professor Taifo Mahmud, PhD from Oregon State University, USA, and Dr. Mirna Maharmi Iskandar. Now, we will give a certificate, placard, and batik to our keynote speaker, which will be given by Dean of the Faculty of Medicine and Health Science, Dr. Humarianto, SOPT MCAS. For Dr. Humarianto, we please you to the stage. And for the committee, we please you to give a help. We're gonna take a picture together. So we invite for Bu Indri, Bu, Bu Umi, Bu Rizka, Bu Putri. Yeah, for all head of department and coordinator of department for each major from medical, psychology, pharmacy, public health, and nursing, we please you to the stage. Bu Puspa, Pak Syamsuriza, we please you to the stage to take a picture together. And Bu Fadliana, Bu Fadliana, we please you to the stage to take a picture together. Bu Fadliana, we waiting for you.